So I will talk about giving a brief overview about plant pathology. And I will try to give you an insight about the cause of plant diseases. I will talk about the different lifestyles of pathogens. I will give you an idea about the innate immunity of plants and how the molecular side of the plant immune system looks like. I will give an insight about the local and systemic responses in plants and the preformed and induced resistances in plants and the built-in growth defense antagonisms which are inside plants existing. And I will talk about coevolution between pathogens and plants. So plant pathology, if you start with the word, comes from the Greek term pathos for suffering or disease and for logos for studying. So we study basically not the healthy plants, but we focus at, for example, this rust diseased wheat. So we study basically the plant diseases in plant biology, plant pathology. And plants are facing all kind of different stresses when they are growing out there. And one major cause of stress are obviously abiotic stresses, which are leading to reduction in crop production, for example, in growth reduction, and of course can lead to complete loss. Also, the quality is decreased of the crops which are harvested. In biotic stresses, basically under biotic stresses, plants are also showing a reduction in growth, but the diseases or the damage can be even more severe because many pathogens are producing toxins, like for example here this Claviceps purpurea fungus causing these ergots. And this is a picture from the Isenheimer altar, showing a diseased person which has eaten too much of this ergots in the bread, and thereby getting burned from inside in terms of its feeling. And there were many people dying of it in the past. So this can, of course, destroy basically this kind of contamination can then really destroy whole mixed crops or whatever you have harvested if you don't select it out. So what causes these biotic stresses? And if you look at the causes, they are manifold. There are insect diseases, biotic stressors, there are nematodes, there are fungi, plant pathogenic bacteria, parasitic plants, herbivores, plant viruses, oomycetes, but also plants fight against each other with allelochemicals. So they are secreting chemicals to outcompete other plants. So there are manifold biotic stresses and causes of biotic stresses. And just to give you an impression about the numbers, what we're talking about, there are 19,000 fungi known which can cause disease in crop plants. There are more than 1,000 plant viruses known to infect plants, or 4,000 plant parasitic nematodes which are infecting plants. So we're talking about different species, and each species has its own strategy and its own niche in plants. So it's really a complex topic, and we will just be able to learn today a few aspects of it or principal concepts of it. So among those pathogens, there are biotrophic pathogens, which are living from a living host, here exemplified by this farmer getting the milk from the living host. And then there are other pathogens which are living in necrotrophic lifestyle, which are living from a killed host. And there's a specific point here that there are biotrophs which are really depending on its living host they cannot live on dead material, as saprophytes. So these are then called obligate biotrophic pathogens, which are really depending in their life cycle completely on a living host. There's another feature. Some pathogens are having a biotrophic phase and a necrotrophic phase, and then they are called hemibiotrophic. So they are having first a biotrophic initial start, and then they go into killing and eat up the dead or the freshly killed plant. And so plants have to differentiate when they get under attack. They need to know, or they need to basically, in order to react appropriately to the specific lifestyle of the pathogen, they need to recognize, am I under attack by a biotroph, or am I under attack by a necrotroph, or maybe of a hemibiotroph. And accordingly, plants are reacting on the molecular level towards these different biotic stressors. So what makes a plant in the end diseased? What causes in the end an outbreak of symptoms which are clearly visible and clearly leading to the term this plant is sick? So we have different aspects. We have the genetics of the host, which is an important part of how a disease outbreak can occur or not. And on the other hand, we have the genetics of the pathogen. And we have the environmental factors, climate, water, accessibility, etc. And in this interplay between these three major components, a disease can either develop or not. So as a farmer, you have quite a control about what you are putting on into the soil in terms of seeds. So the genetics of the plant on that side is more or less in your hand. You have no control about which pathogens are out there. And you have, depending on the type of agriculture or practice, you have more or less control or no control about the environment. But if you look now at the plant and you see this picture, and this would happen to us, that there are pathogens eating up half of our organs, I'm pretty sure we would not make it. We would just be killed. But plants are different. Plants are built up in a modular way, and plants, they cannot run away, but they have a dramatic or a fantastic power to regenerate. So the same plant, after a few weeks, if the pests are taken away, can recover fully, and you will not even see that it had been ever under attack. There will be new leaves built in a modular way, and they can simply replace the destroyed leaves. And so to understand the conceptual difference between plants and animals, I think this one needs to be always aware that plants have this regenerative power. They can afford to lose a leaf without having an impact on their individual life. And that's why most plants, if you look into nature, are looking quite fine and happy. And this is depending on a strong immune system. And the immune system has different layers. We have on one hand a preformed defense or immune system. These are preformed defenses. You have a bark which prevents infections. We have, everybody knows, thorns and spikes. It's basically analogous to a barbed wire. And then we have here, you see these little hairs, trichomes called. They are filled with chemical defense materials. So everybody has experienced this as a child for sure, getting this burning by touching a leaf of this plant. And so these are all preformed defenses. And the preformed defenses are passive defenses. They are not reacting towards a pathogen. They are just there already in advance that potential danger is taken away. You have physical barriers like voxy and cuticular layer on the epidermis, on the outer layer of the leaf. You have cell walls which are protecting. You have stomata and lenticels. These are specific cells or structures which are regulating basically what is coming in and what is going out. And then there are chemical barriers, and these chemical barriers you find, for example, in this wash nut, which you can use as an alternative to washing powder. They are in the seed coat enriched to prevent infection. They have antimicrobial activities. You find them also in oat, for example, here a structure. This is aminazine A1 or the alpha-tomatin. These are all so-called phytoanticipins. These are chemicals which are present in advance of any attack. There are other examples of these phytoanticipins, of these chemicals which are there to prevent infection, which are antimicrobial. You know all these brassicasses, which you are, I guess, eating once per week at least, in one or the other way, and they contain glucosinolates. And these glucosinolates are basically like a mine, like a landmine. And then there are myrosinases. These are enzymes, these are proteins, which can trigger the explosion of this mine. So the moment the myrosinase is removing this sugar moiety here, then you get a uncontrolled reaction happening of chemical reactions, spontaneous chemical reactions occur, which are producing all kinds of highly toxic compounds, which are harming the organism which is trying to eat these plants. And how is this done? If you think of a highly dangerous chemical here, which has the potential to kill any living, this is done by compartmentalization. And this is also, again, a very important aspect if you want to understand the plant immune system, to understand the compartmentalization. So there are cells inside the tissue which contain the glucosinolates, this chemical compound which can explode if you want. And then there are cells which contain the enzyme which can trigger the explosion and the release of the toxic compound. And then if you basically bring them together by a chewing insect, which is breaking through all the compartments, then basically the chemical compounds are released and inhibit basically the digestion of these pathogens, in this case of these pests. So another aspect which is important to understand the buildup of the plant immune system is the relation between growth and defense. And I try to show this here in a little graphical way. You have, let's say, this is the genetic potential a plant can grow in a maximum level with 100% if no disturbance happens. But if there's a stressor, any stress coming in, the plants need to adapt to this stress in one or the other way. And this is basically an investment. And every investment put into the adaptation towards the stress is on the cost of growth. So this is basically then in green seen symbolically how such a growth curve would be. The potential of growth is inhibited the higher the stress is. And at the end, of course, the plant will die and there will be zero growth happening. So this is just to understand that everything which goes into defense is an investment which has a cost in one or the other way for the plant. And therefore, investing at the right time is an important point if you think of a defense system and the immune system of plants. So this linkage between investment into defense and investment into growth is basically what you see also in societies, and you see this also in plants. So the more investment into defense is put, there is a cost on the other side, which is where the money cannot go or the energy cannot float in. And this is just an example to visualize this for you. Here is a plant mutant which is highly induced in its defense responses. And you see it's a dwarf in comparison to the wild type plant. And this is accumulating high levels of a plant defense hormone, which is called salicylic acid. And other pathways are induced strongly. So these plants are highly resistant against bacterial infections, for example, but of course, on the cost of defense. And this will be, again, a cost in a competition with other plants that they cannot afford in the field, because this mutant will not make it in competition to other plants which are not investing everything into defense. So this is leading us basically to the second layer. Beside these preformed defenses, which are existing in plants, we have induced defenses. And this is meaning just at the right time, at the right place, you need certain compounds or certain strategies to avoid penetration of pathogens. And if you see here the list, again, we have structural defenses and we have chemical-induced defenses. We have, for example, the lignification. It's basically some kind of chemical sealing happening upon infections. Callose deposition would be also falling into this group, or the separation of whole leaves by forming abscission layers, which are separating one leaf from the rest of the body. Tyloses, this is basically a filling of the vasculature, the pipe system, if you want, from a plant when pathogens are trying to invade from there, or cork formation to prevent penetration from the outside. So all this, if you summarize it, it's about sealing and restricting. You don't want that something enters further. And on the other hand, you have chemical defenses. You have the production of reactive auxin species, which is killing off anything, basically. Then you have phytoalexins. These are antimicrobial, low molecular compounds small molecular compounds which are showing antimicrobial activities and which are produced just in time at the place of infection. You have specific proteins which are targeting components of the pathogen, for example, chitinases. These are enzymes which degrade the cell wall of fungi. And also cork formation falls partially into the chemical defense. And so this would be chemical warfare, and plants are very strong in this chemical warfare. And last but not least, we have the hypersensitive response, which is that simple host cells are killed in a programmed cell death manner. And I told you already, plants can regenerate. They have a high regenerative power, so they can afford to kill whole areas in their leaves without having a big impact on the whole plant in order to prevent further spreading. And that would be the strategy if you want to find an analogy. When the Iraqi army was leaving Kuwait, they have just burned the oil field so that the Americans will not profit from it for a while. So this is basically the 3 different strategies which you can find in summary in the plant immune system in the induced defenses. Looking at the phytoalexins, these are these antimicrobial compounds which get in time produced. There's just one example here. We have sorghum as a control plant. Here you see nicely green, no colors appearing. But if you infect sorghum with Sporozoium rallianum, then you basically see an accumulation of, in this case, of a deoxyanthocyanidin. And this is a phytoalexin, which prevents further spreading. And you see the very same reaction if you just trigger it by adding chitin, which is a component of the cell wall of the fungus. So this should lead us basically to the next part. You don't need to see the whole pathogen. It's enough to see a part of the pathogen, a piece of it, to already react. So the principle of a responsive immune system is that you have the recognition of danger signals. You need to recognize what is self and what is non-self, or what is unusual self, let's say, for example, a degraded cell wall of your own. And then these signals need to be further transported basically within the cell. You have a processing and the integration of all kind of incoming signals. And then there needs to be made a decision how to react. So the responses can be structural, chemical, there can be developmental responses, local and systemic responses inside the plant. So what is really needed to recognize something? And I would like to show you that also we don't need much. We just need certain patterns. And the moment we see a specific pattern, we already understand what it could be. We don't need the full picture to have an imagination what it could be. And very similar, plants don't need to see a full pathogen. They just need to see a pattern. It's enough for them to see either a pathogen-associated molecular pattern, a pump, or a damage-associated molecular pattern, for example, a piece of their own cell wall. And this will be an alarm signal for them to say, oh, I'm in trouble. I need to start my defense system. So as we just recognize specific patterns and we interpret it, also plants do the same, just with a different way. They have different receptors at their plasma membrane, and they recognize specific molecules which are highly conserved in fungi or highly conserved in oomycetes. And evolutionary for those organisms, to lose these compounds would be such a fitness cost that they cannot afford it. And that's why the organisms or the plants have focused on those molecules and patterns which the other side cannot avoid without losing its fitness. Humans or mammals and plants have some commonalities here and also some differences, and I would like to show you this here. So plants have these pattern recognition receptors at the plasma membrane, and also we do have these receptors which recognize pathogen-associated molecular patterns. Just the number is much, much lower. So in mammals we have few, in plants we have hundreds of them. And also the localization is a little bit different, but this is not important here. Another very important difference between a mammal immune system and a plant immune system is that plants can only recognize patterns for which they have a receptor hardwired encoded in their genome. whereas mammals, they can evolve recognition of patterns which they have never seen in pathogens before. This is how our immunization works. And so this principal difference needs to be understood to understand why we need also genetic variation in crops, for example, because only this way we get robustness against certain pathogens. And so here, another commonality here is, and this is interesting, we have convergent evolution. We have basically patterns which are recognized by receptors of plants as well as by receptors of animals. But this is not because they have evolved from the same source. They have basically convergently evolved because these patterns are so specific and so unavoidable for the pathogens that they are simply perfect patterns to recognize those pathogens, like flagellin. So this is, should show you now a plant cell, and these are these receptors at the plasma membrane, the perception system basically in plants. And if a pathogen is landing on the plant, it's automatically, or not automatically, it will be recognized with the help of these receptor-like kinases. And this is then triggering an oxidative burst and a pump-triggered immunity response. And any pathogen which wants to be successful to invade its host needs to suppress this recognition or the signaling cascade. And now we come to a second layer of a warfare or an evolutionary war between pathogens and plants. So pathogens have evolved over millions of years tools to secrete them into the plant and to suppress these signaling cascades. And this is done with the help of effectors. These are secreted molecules from the pathogen which suppress in one or the other way this response. And this is then, again, a new, let's say, starting point for evolution, because now there are molecules coming from pathogens, and plants have evolved then receptors, which are called NLRs. And these receptors, again, recognize effectors, either directly or indirectly. And upon recognition, this leads then to a very dramatic response, because these effectors are indicating I'm really heavy under attack. It's not just a bacterium on my surface, it's a bacterium which wants to manipulate me because it's secreting effectors into me. And when this is recognized, then the reaction of the plant is very strict. It's basically leading to an effector-triggered immunity response, which is nothing else than a programmed cell death. And this programmed cell death, again, would be like when the Russian army was escaping the Napoleon attack and Moscow was burning. So basically you leave nothing behind, it's all burned, and the pathogen has no advantage of the place. So if you look at this coevolution of having recognition and overcoming recognition with the secretion of effectors, this has led to the famous Zig Zag model in plant pathology, where you have basically here an evolutionary time course, and you have here low immune reaction and high immune reactions of the plant towards an incoming pathogen. And initially, you have a recognition of the pathogen, or the pumps the pathogen-associated molecular patterns with these receptor-like kinases, which is leading to pump-triggered immunity. Then there are effectors coming in, suppressing these responses, and you get effector-triggered susceptibility. New receptors are formed, the NLRs, causing effector-triggered immunity. New effectors come into game and suppressing this again, and so forth. And this is an endless war. And this is why every, let's say, pathogen will overcome any resistance at some point in evolution, in an evolutionary scale, because there's simply, it's an ongoing war, and it's very difficult to basically break this completely. It's simply it's a tango between the pathogen and the host in an evolutionary aspect. And this has led also, or this model was evolving from an understanding that there are, on the plant side, resistance genes, here R genes, which are recognizing on the pathogen side avirulence genes, which are basically effectors which get recognized. And this is here the gene for gene model, which has been first built up by Harold Henry Flor, where he has basically shown that if there is a resistance gene and on the pathogen side the corresponding AVR gene, you have a recognition and the plant stays healthy. But if either the resistance gene is missing or the effector which is getting recognized is missing, then you have infection happening here for the rust in this case. But this is only true for biotrophs. In case of necrotrophic pathogens, there is a different strategy. Necrotrophs want to kill. So necrotrophs have factors which are necrotrophic effectors or necrotroph effectors. And these effectors, they need something on the host side to be active, a susceptibility gene. So here it's basically inverted. That's the inverse gene for gene model, where you have an effector which needs a susceptibility gene on the host side to be active. And then you get infection and the cell death happens and the necrotrophic pathogen can spread. Whereas if the host susceptibility gene is missing, the effector cannot act, and so pathogenicity is low. And if the effector is missing, of course, then also pathogenicity is low. So this is just to give you some model. And in both cases, basically, for the biotroph or the necrotroph, as I indicated already, plants need to somehow specifically respond. And also, although this is very schematic, biotrophs are basically recognized and then triggering salicylic acid defense signal responses on the plant side. And necrotrophs are usually recognized and triggering then jasmonate and ethylene signaling responses in the plant side. And then there are known marker genes, and the promoters will still will be necessary to understand why we build reporter lines. The smart plant concept comes from this concept here, that basically we can differentiate by the marker genes which pathway has been taken. And of course, in case of the wiring inside the plant, the salicylic acid signaling prevents the JA signaling pathway, and the JA signaling prevents the salicylic acid signaling pathway. And this way you have a certain channeling of resources in the plant. So this recognition and the responses are not only local happening, they occur also systemic. You might have a leaf getting infected, you have local responses, but then signals are transported throughout the whole plant and warning basically the other tissues, take care, there is an attack ongoing on the body. And this signaling is called systemic signaling, or a system. Yeah, we will come now to this. And actually, by the way, there are also signals which are volatile and go from plant to plant. And it has been shown that basically plants can even warn other plants, even not from the same species, if they are under attack. So there are two principal ways of systemic resistances or induced resistances. There's the induced systemic resistance, ISR, which is triggered, for example, by rhizobacteria. And this is then leading throughout the plant to an enhanced, let's say, responsiveness of the plant towards an upcoming pathogen attack, and shows very little, let's say, growth trade-offs, which is basically nice from the farmer's point of perspective. And then we have the systemic acquired resistance, which is salicylic acid driven, which is triggered by pathogens, and there the growth trade-off is much stronger, and leading to a different setup of genes, PR genes going up, etc. So based on this, and I indicated this already before, just to point out what our PhD student financed by PhenoRob is doing, she is building basically reporters to basically create plants which can then be visualized, or which easily visualize an early infection already by having a colored reporter, which is leading to this beta-lysine accumulation and indicating then if it's, for example, an SA signaling pathway or a JA signaling pathway, indicating it's a necrotrophic pathogen or a biotrophic pathogen infecting this plant somewhere. And these plant reporters are under progress. Another important aspect in the plant defense system is the compartmentalization of defense, because the less let's say if you have it at specific places at the borders, for example, a chemical barrier here, like here in this root, which has been cut through, and this is a root of oat. And you see here this autofluorescence of a chemical compound, which is avenacin A. It's, as I said, a phytoanticipin, and it's preventing, basically, it's like an antimicrobial barrier that pathogens cannot just cross easily. You see also the glucosinolates are not produced evenly across the whole tissue. You see here a promoter reporter line for glucosinolates related gene biosynthesis genes. And you see here that they are very specifically expressed in, for example, the vasculature or at specific places in the root and not everywhere. Again, it's about expression or production of the compounds at the right place, at the right time, and not just everywhere in order to prevent growth defects or growth loss. Part of this organ and cell type specific defenses is also the stomata. The stomata are basically the guard cell formed by guard cells, which are regulating the incoming air and outcoming air flow for a leaf in order to breathe and to take up CO2, but also for the evaporation control it's needed. And this is an entry point for bacteria, as you can see here. So stomata these cells have a very unique and specific defense system, which is different from the neighboring cells in terms of the outcome. And this is here seen if bacteria, this is a graph showing you either a leaf in white treated with water or a leaf treated with bacteria, which are Pseudomonas in this case, infecting plants. At the very beginning with water, there is no response. With the bacteria landing on the stomata, they close directly. They recognize I'm under attack, I close my gate. And because these are pathogens, they have effectors and they have tools to open them up after 4 hours. And I just would like to point out here, this is a control where they used E. coli instead, and the stomata close, but they stay closed even after 8 hours because the E. coli don't have the effectors to open them again, the stomata to get into the leaf. Also, just a side note, also fungi get recognized via the chitosane, but this is just basically the same in different colors. Also, another example for an organ-specific defense is the root tip here. You have, in plants, you have the so-called, you have basically cells which are released from the root tip, which are loosely there, and they are needed to lubricate basically the passage of the root tip, which is the place where the cell division occurs, through the soil in order to prevent any harm to it. But these released cells, these outer layer cells, which are released and which have no direct connection with the plant body, they have additional functions. They have the function to attract actually pathogens that they get infected, and by this they prevent infection of the real plant body, so misguiding them. There is a defense matrix built up, so they release all kind of long sugars and long chain sugars, as well as extracellular DNA. They have antimicrobial root exudates are occurring here from them to kill basically just by direct compounds. And so this is how it looks like then in a real root, if it's this is colored here, and in red you see basically these root extracellular traps, which are composed of sugars and extracellular DNA, which are like nets in which the bacteria here in blue get trapped, and they cannot go further towards the sensible part of the root, to the root tip where cell division would occur. So again, plants need to invest their defense at the right time, at the right place. And so pathogens know this and they exploit this, because these are mechanisms which are built into the metabolism. And so pathogens try to push the growth, or certain pathogens try to push growth, uncontrolled growth, like for example Osteoargomedes, in order to reduce defense. So there are many, many pathogens, and this is just the classes: viruses, bacteria, protozoa, chromista, true fungi, parasitic plants, rotifers, nematodes, insects, mites, which cause galls and tumors on plants. And the reasoning behind could be that on one hand, they create a sink tissue where sugars are floating in because these galls are not anymore photosynthetically productive. They are sink tissues like roots where the sugar flows to. Furthermore, by removing basically any kind of tissue specificity, also the loss of cell type and tissue specific defense reactions occurs because it's a redifferentiation happening here. And it's also forming a niche for the proliferation. So this is why pathogens like to push the growth pathways in a way, in an uncontrolled manner, in order to basically later reduce the defense. So coevolution is really driving, as I showed you now here with this growth and defense antagonism as well, but I showed you also other examples, is really driving the plant pathogen interactions. And I would like to show you a few last examples here. So a chemical warfare is happening here between Alternaria and Brassicaceae. And Alternaria brassicicola is a necrotrophic fungus causing this spotted disease here, for example. And the plants produce a phytoalexin, brasinin, which is inhibiting Alternaria brassicicola. But Alternaria brassicicola so it's an antimicrobial compound from the plant. And this fungus has evolved an enzyme, brasinin hydrolase, which is degrading brasinin. So plants again produce cyclobrasinin, which is inhibiting in a competitive manner this enzyme. And again, evolution moves on. Alternaria had produced or evolved a cyclobrasinin hydrolase, which can again attack this compound and degrade it. And plants moved on. They have evolved additional phytoalexins, camalexin, brassilexin, which are inhibiting again this enzyme. And this is basically explaining also why plants are not producing one compound, one antimicrobial compound. They are always using a whole complex mixture of compounds because, first of all, this way they prevent that certain enzymes on the pathogen side can still act. At the same time, it's also like a precaution. They are producing just a variety of many different chemicals to prevent the spreading. Also, in case of the glucosinolates, there are such kind of co-evolutionary processes ongoing. For example, here, this diamondback moth is needing for its larvae Brassicaceae. They are specialized for Brassicaceae. And in order to detect where to place their eggs, they are smelling the glucosinolates, which are supposed to defend. They are smelling them, and this is for them the indicator where to place their eggs so that their larvae, which are resistant towards the glucosinolates, can grow and live happily and feed. Or another example would be, for example, this little Pyrrhus rape, which is supposed to be killed by the action of the glucosinolates if the isothiocyanates would be released. This is the chemical compound coming from the glucosinolates. But this little, the larvae of this little Pyrrhus rape, they are producing an effect, basically an enzyme, nitrile specifier protein, which basically blocks this chemical reaction. It's not occurring anymore. And instead, nitrile is formed, which is not harmful to the larvae, and therefore they can grow and happily feed on these brassicaceae. And last but not least, sometimes the defense system becomes the own defense system. For example, here this aphid is feeding on Sinapis alba, and this would be the natural pathogen, the natural control agent here, eating up the ladybird beetle larvae are eating heavily, usually the aphids. But when the aphid is eating the brassicaceae, it's accumulating here the glucosinolates within its own body in a specific compartment. And at the same time, while sequestering the glucosinolates coming from the plant, it's producing its own myrosinase. This is the trigger to make them explode if you want. And this leads then to the death of the ladybird beetle if they start eating the aphids. And with this, I'm Basically, at the end, I would like to summarize my take-home messages that we have a disease triangle composed of the genetics of the plant, the genetics of the pathogen, and the environment. We have always ongoing a co-evolution between plants and pathogens, and this is just a moving target here. Plants have an innate immune system. This means it's genetically encoded what they can recognize and what they cannot recognize. And if they cannot recognize it, they cannot learn it, as we do. And plants have preformed resistances as well as induced responses towards pathogens. The defense is widely a chemical warfare beside the structural defenses. And plants react local but also systemic. And the tissue-specific and stage-specific responses are also occurring in the plant immune system. And with this, and most important take-home message, I think, should be that plant-pathogen interactions are fascinating. And with this, I thank you for your attention, and I'm open for your questions. Thanks.